What comes to your mind when you think about the immune system? Now think about Africa. What does it bring to mind? On ImmunoAfrica, we invite professors, postdocs, PhD students, and everyone in between to tell us about their journey into immunology, the questions driving their current work, and the impact they hope to make on the continent. I am Wealth Okete, your guide on this adventure, or better yet, your co-explorer. In each episode, I bring you the inspiring stories, innovative approaches, and invaluable perspectives of the Africans advancing the field of immunology, both locally and globally. Join me for an exciting adventure into immunology through the hands and eyes of Africa.
The third edition of our graduate student series spotlights the stories of 5 African PhD students coming from Zimbabwe, South Africa, Benin, Cameroon, and Ethiopia. Our guests are leading research projects to answer fascinating immunological questions across HIV, tuberculosis, parasitic infections, and keloids.
Limpho Thipane is our third guest. She is currently enrolled for a PhD at the University of Cape Town, South Africa, where she studies tuberculosis in the central nervous system, CNSTB. Her research aims to unravel host pathogen interactions and the role of immune cells, such as microglia, in CNSTB progression and control.
My love for science started back in primary school, actually. So we have a subject called natural sciences, right? So that's where we start learning about plants and how plants grow, about many things, specifically like nature, right?
Mhm.
And then I was like, okay, this subject is something that I actually enjoy more than anything, right? Okay, cool. And then after that, I went to high school. And then that's where we did life sciences, right? Or biology. So that still sparked an interest in me. I was like, okay, I'm enjoying this more than other subjects, right? But I also enjoyed accounting, surprisingly. And then after high school, I think in grade 11, that's when they started teaching us about the heart, right? The anatomy of the heart. And I was like, okay, I'm actually loving this. I'm interested in what's deoxygenated blood. Why is the blood blue? Why is the blood red? What comes where and why the heart goes, like the blood goes there, things like that. So that actually made me want to know more in terms of what happens in the human body. Grade 12, we learned about the brain. Now that's where I became really obsessed about neurosciences, but I didn't know at that time that it's neurosciences. So they taught us about the brain, and I was like, okay, the brain is a very important thing. Yes, it can control the human body, but it also can control your emotions. Now that's where I went to university, and then I did psychology and physiology, which was like a combination of both, right? But it was mostly focused on the brain. So it was a win-win for me, right? So I did that, and I enjoyed physiology more than psychology. And then I went to UCT, where I was accepted for my honors for neurosciences and physiology. And then that's where I saw that you can actually combine neurosciences and immunology. And this is where neurosciences and immunology has led me.
Can you recall one of the fascinating or surprising things you first learned about immunology in the context of the brain?
I actually learned that there are so many cells that people don't know about, and those cells, they also have like important roles, right? But as much as the cells are important and they are always there to protect the brain, they can also be bad. So they can be good or bad depending on the severity of the pathogen. So for example, like TBM, we have microglial cells, right, which are known as the first responders to injury or infection. But if they can't control the bacteria, they start becoming bad. So that kind of like worsens the pathology of the disease. So that's one thing that I've learned. And I've learned that like there's a lot of cytokines and chemokines. They have different names, they have different functions. So that was overwhelming for me at first. But I mean, like science is something that's always evolving, and we learn as time goes on. So yeah.
Yeah. So I think from what you said about the microglia and the roles they could play in brain diseases or brain-related pathologies, It's just that, when we think about the immune system, usually like the good guys versus the bad guys, how the good guys are trying to just reduce the number of bad guys and kill the bad guys. But your
Mm-hmm.
work is making us see that good guys can also turn bad guys sometimes, right?
Yes. Yes,
Yeah,
yes, yes,
yeah, yeah,
that's true.
yeah. Take us through some of the earlier projects you did in the context of the brain and the immune system.
Okay, so in my honors, we wanted to do or mimic the CNS, right, the central nervous system of the brain. So we wanted to do cultures, right? So we were not able to do that. So what we did was we started using mice now. So we started involving mouse work. What we did was we looked at microglia cells as our prominent cells in the brain, and we looked at using a diet that actually depletes microglia so that we can see if there's no microglia in the brain, how would the brain respond to the TB once the TB gets inside the brain. So this diet is called the PLX 5622, which is a formulated rodent diet provided by the US. It depletes or silences microglia cells depending on how long you actually give the mice this specific diet. So once the mice start eating the diet, it actually silences the microglia, and then the most prominent cells that are there is your astrocytes. So your astrocytes are cells that are important for preventing injury spread throughout the brain, and then we also have neurons, which are your messengers within the brain. So we would infect mice with TB and see how the CNS actually responds, the microenvironment responds to the TB. So, but for my honors, we just established that mouse model. And then for my masters, that's where we started infecting mice with TB. We also did an infection with BCG, GFP, which is a attenuated strain of TB, so a less dangerous, strain of TB. And yeah, we actually found interesting results.
Yeah.
Yeah.
Yeah. So I think before we start unpacking the results, the fact that you're looking at TB, which is everybody knows TB to be a lung disease. If you have TB, you should cough, your lung is weak, a lot of things can happen to your lung, but then you're looking at TB in the brain. So let's just try and unpack the fact that TB can actually infect the brain. And while unpacking that, I also want you to address the question of, were you injecting the bacteria into the brain, or were you just injecting it into any part of the mice, and then the bacteria would make its way to the brain by itself?
Okay. So the first question is TB can infect the brain, right? Okay. So we know that TB is the primary site of infection is the lungs, right? Or the lungs. Yeah, they are the lungs. And TB is known as a multisystematic disease, meaning it has abilities to actually infect other parts of your body, right? So your kidneys, your spinal cord, and eventually your brain, right? So TB, once it has caused damage to your lungs and there's no way of fighting the TB, it can actually escape its way into the spinal cord. And remember, the spinal cord is also connected to the brain, right? So once the bacteria travels to the brain, there are many theories on how the bacteria actually crosses the blood-brain barrier. So the blood-brain barrier is a structure that is known to protect the brain. So TB actually has the ability to actually go inside the brain. So yeah, that's that. And then how did we mimic CNS TB, right? So there's a procedure called intracerebral infection where you put the mice under anesthetics and then you shave the head, you make an incision, and then you actually infect the brain with TB directly. So that's what we did. And yes, it's ethically approved.
For that study, you were trying to deplete the number of microglia cells in the brain and then see how the mice will be able to respond to TB in the absence of those cells.
Yes.
So what
That's
was
exactly
the
what we're trying.
major finding from that before we proceed to your master's project?
So we actually found that this is actually my master's project, what we did. So we actually found that by us giving the mice this diet, right, the PLX 5622, it does not only deplete the microglial cells, but it also depletes other cells that are involved in the immune system, right, in the CNS immune system. So we found that it also depletes macrophages as well as dendritic cells. So we know that dendritic cells are also important in terms of responding to injury or infection, right? So when I went through literature, it actually said that some of these cells are associated with the receptor that this diet actually silences. So that was actually interesting. And a lot of studies have done something like this, but it's always been in Alzheimer's or other neurodegenerative diseases, but not in CNS TB before. So that was actually cool.
If we were to translate some of your findings, or maybe not translation might be like too
A big, yeah.
yeah, a very grand dream. What relevance do these findings hold for TB in the brain or neuro TB in humans?
So the thing is with the drug, right? Studies have shown that, especially in mice, studies have shown that the diet or the drug does not cause any behavioral changes. So that is a positive thing, right? And it also helps in terms of like taking down the bacterial load in the brain, which is also a good thing, right? But remember, we use mouse models to try and mimic what happens in the human body, right? So we wouldn't know how the humans would actually react, how the human immune system would actually react to the specific diet. So maybe you'd find that in humans it might be detrimental, but in mice it's actually a positive thing. So it would be nice though to actually try and see if this can help in terms of like decreasing the CNS TB cases because it's really difficult to actually diagnose it. And once it's been diagnosed, you'd find that it's too late now to give the certain TB regimen for that person. So yeah, it would be interesting and it would be a dream actually to try and translate results that I've gotten into humans. But again, such things they take time and they also need to be tested out to ensure that we are not causing any harm to humans.
So the microglia cells, you were depleting them because they contribute to TB pathology in the brain.
Yes, so microglial cells are known as the first responders, right, to injury or infection, right? So once they feel like they can't control the infection or the injury, they become activated for a long time. So uncontrolled activation of microglial cells can actually cause neuroinflammation, right? And neuroinflammation is not good, right? So that can also worsen the bacterial spread throughout the brain. So that's what we wanted to actually prevent. So because we know that TB is a very dangerous and it's a very uncontrollable bacteria. So that's what we wanted to actually eradicate or understand what would happen in CNS model if we deplete microglial cells.
So I wanted to ask because the podcast is concluding a TB immunology series right now, where we are speaking with like professors and postdocs who work on TB vaccines, TB antibody responses, TB risk signatures, and several aspects of TB. And I think something I've learned from speaking with all the scientists is that TB is a very heterogeneous disease. The expression, the manifestation in different individuals is different. In individuals differ. And I'm just wondering because the emphasis in the series were just lung TB, but this is brain TB, and I know probably the brain is probably more difficult to study than the lung. But do we know so much about the manifestations of TB in the brain? Is it different among different individuals? Is it do different TB strains lead to different manifestations of TB in the brain? And what do we know about the heterogeneity of brain TB?
Okay, so central nervous system, right? It consists of three forms of TB or CNS TB. So the thing is, with such manifestations, it's difficult to actually try to treat them, right? Because different forms of TB requires different types of TB regimen, right? Because it will be a problem if you start treating something, but you're actually not treating like you're actually not giving the patient the right treatment, so they can actually worsen the condition. So I feel like more research needs to be done in terms of like finding first of all, medication that can actually pass or go through the blood-brain barrier, and more diagnostic tests or like approaches in terms of how doctors can know which kind of TB they're treating for. Because I feel like if they don't treat the right one, that also increases the incident cases as well, which is a problem. Yeah, because people just know that TB is a lung disease, but they don't know that it can actually go to other parts of your body. So that's where the confusion comes in.
Yeah.
Yeah.
Yeah, I think there's been so much emphasis on lung TB that we do not realize that there's TB in other parts of the body. So for your PhD, I know you just started, but what's the big question you're trying to solve with your PhD?
Okay, so my PhD is actually something that I'm so excited about. It's something that's new, but I'm so excited. But it's also overwhelming because it's a new thing, you know. But it's also something that I've been doing as well. But basically, I'm looking at how, do you know cannabidiol, CBD, right? It's part of the yes. So what we want to do is, according to research, it has proven that it can actually reduce inflammation, right? So they've shown that it can actually reduce inflammation. So we want to see if that can actually work in terms of TBM. So TBM is tuberculosis meningitis, which is also one of the forms of CNS TB. So we want to give the mice this CBD and then infect it with TB and see if that can actually decrease the neuroinflammation that happens during TBM. Yeah, so that's what we want to do, and we also want to look at the immunology part of it, but obviously using flow cytometry, which is a famous thing that we do here in science. Yeah, flow cytometry. We also want to use imaging as well to see if the bacteria has crossed the blood-brain barrier and if we can see the bacteria in the brain. And yeah, that's basically what we want to do. I don't want to spill too much because it's something that's new, and I'm also
So basically, you're looking at CBD and TB inflammation, brain TB inflammation.
Yes.
Yeah.
Yes.
Looks really.
That's me. Neuroscience, neuroinflammation. That's me.
Yeah. So you are involved in a whole lot of things. I think reading your profile, I want to ask, how do you manage your time? How do you ensure that you're able to coordinate all these things and still have time to study neuro TB?
I feel like it's just a thing of you make time. Like you make time. You need to prioritize things that you put yourself in, right? So with school, I know that okay, I need to go to school, I need to do this, I need to be in the lab, and I need to get things done, right? But if I know that there's something I still need to do, I actually live by like a to-do list. So every day I'm like, okay, by the end of this week, I should be done with 1, 2, 3, 4, 5, you know? So I try to manage my time effectively and efficiently, but also I don't deprive myself from living, basically. I don't make I don't make school my life. I don't make this my life. Like I just want things to actually coexist in a way, just so that I don't overwhelm myself because I enjoy what I'm doing. I enjoy giving out to the community. I enjoy spending time with the students. I enjoy science. I enjoy being in the lab. So everything can coexist. So yeah, that's what I do to make sure that everything doesn't overwhelm me. But I do have my days where I'm just like, maybe having a lazy day wouldn't hurt. So I need to rest, you know?
Yeah.
So yeah, nobody's perfect. Yeah. So I also have a lazy day here and there. Yeah.
So I think something that's striking from what you said to me is we have to make time for the things that we get ourselves into. So you decided to do all these things. So
Exactly.
you have to make time.
Exactly.
If you can't make time, please step down and focus
Please
on
give others a chance.
I've heard people say that if you are procrastinating, and when people procrastinate, they're supposed to do something now and then they're doing something else. They said that's something else you're doing. Maybe that's what you should just focus on instead of if something is important to you or compelling, you should be able to make time for it. If you're not making time for it, maybe you have to rethink how much value it's bringing you. Yeah.
Yes, definitely, yeah.
Yeah. So I've heard you speak and talk to these students about science, about immunology. I think one of the AWSA workshop when you taught them about TB, I was like,
Yes.
Dimpo is an amazing communicator. So I think you need to
What?
share. Yeah, yeah. Because I think I did not. I'm not saying it because you're here, of course. But one
Mhm.
skill in communication is you're able to, fit in different contexts into one thing that you're trying to describe. So how do you do that? And what tips can you share that has? I believe even your communication skills would have evolved over the last over the years or over time. So what have you learned,
Mhm.
and what tips can you share on communicating effectively?
Okay, thank you so much for the compliment. So to be honest, public speaking or communicating is a very difficult thing, especially when English is not your home language. But one thing that I can say is if you know the work, you need to be confident and trust that what you're studying is something that's actually impacting other people's lives. So sometimes I get so overwhelmed with like all the knowledge that I have, and I just end up sending voice notes to my friends, and I'm like, oh, actually, this is what I learned today. This is what I'm doing. So like I have friends that are actually interested in what I'm doing, which is something that I actually appreciate because that also allows me to be able to break down scientific and difficult terms
Mm,
into understandable things, you know?
mm,
So in terms of like communicating science to students, it's something that I enjoy because I know that some of us or most of us grew up not having such things. So we only knew about these things once we were older and once we were in university, you know? So tips that I can give is try to be as confident as possible and don't overthink too much. Remember that you know your work and believe that what you're doing is actually it's a good thing. You are trying to improve people's lives and people that are there, they're there because they want to listen to you, not to judge you. And one thing that I also learned is that so in the beginning of the year, I went to a conference, right? The IDA conference. And one of the mentors said that, can you please explain to me your project as if like I'm a 5-year-old student? And that was really difficult. And I was like, okay, now I need to learn how to break down scientific terms so
mm,
that people can actually understand because not everyone's a scientist, not everyone knows what TB is. I'm like, okay, we know that TB happens in the lungs, but people don't understand the causative agent, which is Microbacterium tuberculosis. People don't know, chemokines, cytokines. So I just had to sit down and do like a self-introspection and be like, okay, if you want to make sure that your research is actually known and people understand what you're doing, make sure that you explain it in a way that someone that hasn't been in the lab actually understands. So that's one thing that I'm constantly learning
mm.
how to communicate what I'm doing to people that have never set a foot in the lab. So yeah, that's how I try my best to be a good facilitator.
Yeah. Well done, well done. So, I think you'd mentioned confidence as the first thing, right? Because I think
Yeah.
that which is very important. Yeah. So we should be confident, and we should know that what we are doing matters. And I
Mm,
think even
mm.
from what you shared, would also be open to growing and learning. Like you don't become a great communicator in one day. You have to
Yes,
be
yes,
like,
yes.
yeah, exposing yourself to challenging situations that will just make you, communicate better. Yeah.
Yeah, yeah,
Yeah,
yeah, that's
yeah.
true.
Thank you.
Trust
Thank
me,
you.
even presentations, they're not easy. They're not easy. So, but you learn because
Yeah,
it's
yeah,
either it's either you don't pronounce a scientific word correctly and then someone corrects you during the presentation, or you're not saying something that makes sense. So you learn as time goes on. But
yeah,
yeah.
yeah. True. Yeah, true. And I think kind of this phase of our life being graduate students just gives us that leeway to learn, right? And
Mm,
make the mistakes that can be made now, such that, yeah, we have that opportunity to grow and become better at what we are doing or what we are trying to do. Just take us through what you do, some of your public engagement work, science communication work. Tell us more about them and what kind of inspired you to be involved with them.
so it was a to be honest, it's something that I really enjoy. Like I get excited when I know that there's a workshop coming. So my role there is to basically like ensure that everything runs smoothly in terms of the workshops that we have planned out throughout the year. I select speakers or presenters for different types of topics. It really does not matter what your research is on as long as you feel like you can communicate the science. We have slides that are available. that they can actually be sent to the presenter, and then they present the work, and then we break into groups, and then that's where we start our group discussions, right? So you kind of like understand how the students actually absorb the information, or what they understand from the presentation, and what we as a group, what we can actually improve in terms of like research or like information that they want to know, because they are literally in the rural areas, and sometimes it's difficult for them to even translate information, because you'd find that the pamphlets that are there, they're in English, right? And that also becomes a language barrier. But if we meet the students halfway, we can mix isiXhosa and English, and they can take that information and take it back to the community. So that's the main goal, and it has a very huge impact, right? So that's the IWOSA part. And then in terms of the African Research Society, to be honest, I feel like this year was a year for me to actually get out of my comfort zone, because I felt like I was stagnant, right? And I didn't like that. So I was like, okay, I need to do something that shakes me or moves me. I want to see improvement, or I want to see something moving, basically. Right. So I joined the African Research Society, and I was like, okay, the portfolio that I want to do is the social responsiveness portfolio, because again, I love engaging with people. I enjoy doing things for the community and reaching out. So I got the position of being the director of that portfolio. And some of the events that we have done or are currently doing, the first one was the Future Scientist Day, where we invited students from Tandukulo High School, which is just here, nearby UCT. So we invited like prominent researchers, like Professor Klumani, Professor Elizabeth Main, which is the HOD of immunology. We also invited Professor Sadesh, actually. So we also invited a master's student, just so that they can also hear a different perspective compared to like hearing from professors and as well as from a student. So that event was mainly on ensuring that students should know that there are so many opportunities out there in terms of science. We all don't have to be teachers, we all don't have to be doctors, we all don't have to be, like nurses. There are so many opportunities out there in terms of STEM, right? So the students came, we had a talk, the students asked questions. After that, they went to the lab, just so that they can have a feel of what it feels like being a scientist. They went there, and they had like a practical where they saw like bacteria under a microscope. They used microscopes. And after that, we also took them to the pathology learning center, where they saw cadavers and human bodies, and that actually sparked like an interest in most of them.
I think you're just serving as that bridge between science and society, and it's
Yes.
yeah, it's really beautiful. And the fact that,
Thank you so much.
yeah, I think from when you were speaking, something I was just thinking about is a proof that you've communicated effectively is that the person you communicated to should be able to share what you've told them.
Yeah, so
Yeah,
I hope
because
that you understand what I've been telling you. So
yeah, I do. Even without me, I think even those students you speak to at it was workshops, right? I think with the energy with which they respond, they sound like people that will be able to tell their friends some of the things you've shared with them. Because I feel like that's the whole idea of bringing yourself down to a grade 10, a grade 11
Yes,
students level,
definitely.
right? And speaking with them, because
Definitely.
what their friends tell them, they can tell other friends about it. But what a professor tells them, if the professor tells them that thing as a professor, they probably feel like, okay, do I really understand
Yeah,
this? Yeah, will it
yeah.
matter to my friend, or matter to my classmate or contemporary? Yeah.
And the interesting thing is that I'm not like this Xhosa speaking person. So my home language is Sesotho. So I had to learn isiXhosa just so that me and the students can actually understand each other. So yeah, that's one thing that I'm also like learning, just so that we can try and bridge or break the language barrier, just so that they can also feel comfortable in terms of communicating what they understand. So yeah.
Yeah, well done, well done. Yeah, talking about learning, I would like you to share maybe one of those mistakes you made in the lab that that still maybe haunts you till tomorrow.
Oh my goodness. So, I'm a Harry Potter fan, right? I love Harry Potter so much. So what happens is when we do cultures, right? We take the brain, right? And then we generate single cell suspension. So single cell suspension is when you take the brain, then you kind of like mash it into sort of like we called it like the brain smoothie in a way, right? So you mash it up and then it becomes sort of like a liquid type of thing. And then for you to culture it, it needs like everything needs to be done in the hood because obviously things get contaminated and that's not what we want. Right. So and that takes about 4 weeks. So a whole month, right? Yeah, a whole month. So what happened was I was busy talking about Harry Potter. And then I got so distracted and I opened my cells outside. So my co-supervisor was like, what are you doing right now? And when I looked at my hands, my samples were open. So I had to discard everything. So all like the 4 weeks of work went down the drain just like that. So I
Because
had to
of Harry
repeat
Potter.
again. Yeah, because of Harry Potter.
Aish.
Because of Harry Potter. So yeah, that happened. And then another thing that happened was I was preparing myself for flow cytometry actually. So instead of just discarding the supernatant, I also discarded the cells. So I thought that okay, since the brain has a lot of cells, I'm sure there's something in there. So I just added FexBuff and I was like, okay, let's just go and acquire. We'll see. Maybe there's something in there.
Yeah.
We go to the Patesa, we run the samples. My co-supervisor's like, why is this tube clear? And I'm like, I also don't know. I followed the protocol. I don't know what happened. But deep down, I know that I discarded the cells as well. So I couldn't like explain that, oh, I actually discarded the cells. So that's why you are not seeing any events.
Yeah.
So yeah, so those are the two things that happened that
Yeah.
yeah,
Part
that
of
still haunts me
part
till today.
part of the process of getting
Yeah.
a PhD or getting a master's.
Yeah, masters, yeah.
Yeah.
I mean, like the results won't make sense, but you cook up a story. That's the thing. That's one thing that I've realized in science. You can cook up a story and convince people that this is what you got and this is what it means. So yeah.
Yeah. I think it's always about explaining what the result is telling you, right?
Mm,
Yeah.
mm.
Yeah.
But also, I think it's difficult as well to discuss your results. Some people say it's easy, but I feel like it's a bit difficult, especially when you are discussing something that hasn't been done before. So you're trying to find literature that kind of like backs up what you got, even though
Mm.
it's like different diseases and pathogens and stuff. But using people's research to back up your own research, it's something else.
Yeah, well, I think that's where some of the growth happens because you have to try and make sense of what is happening. But
Mm-hmm.
of course, sometimes it will be difficult, right? So I think maybe another task we could do is uh we started out talking about microglia. So I would like you to spell microglia backwards.
Microglia, right? Microglia. It's
Mm-hmm. Mm-hmm.
AILGORCIM,
Okay.
microglia. Yes.
Okay. Well done.
Those are like my besties. I call them my besties because
Oh,
my master's was mostly on microglia.
yeah. So you're mastering microglia.
Master of microglia, I feel like that's a big deal. Like it's a big title. But yeah, I know microglial cells. Yeah.
Yeah. Yeah. So thank you so much, Dimple. I think it's been a great chat so far. And thank you for the energy and excitement with which you shared your work and for taking us through your journey and your aspirations for the future, your interest in science, communication, engagement, and your
Hmm.
passion for inspiring the next generation of scientists, neuro-immunologists.
Thank you so much for having me on this podcast. I feel so honored actually, and I like the fact that you wanted to hear my story and you actually wanted to see things or like see TB in a different perspective. So I really appreciate that.
Many thanks to our guest, you, our listener, and everyone behind the scenes who made this episode possible, particularly team members Cedric and Cyril. If you enjoyed this episode, be sure to subscribe and leave a review on your preferred streaming platform. Also, feel free to continue the conversation with us on social media. Tag our handles at ImmunoAfrica_ or the Immunology in Africa podcast. Support our efforts to amplify African stories of immunology by nominating a guest or buying us coffee. Visit the link in the description or send us an email, immuno.africa@gmail.com. See you on the next episode.